increasing your immune system to be very, very active. That's basically the fundamental function. And a very active immune system is able to fight cancer. So basically, you put something to a body that raises your overall immune system, and then chance that they fight the cancer cells is very high. One of the very, very high side effects of this is if you have a very high functioning immune system, it can go over the top very soon, that your immune system is overworking and then it fights also healthy tissues. So usually with these cytokine therapies, there are a lot of side effects that actually get a lot of inflammation throughout your body. And this is not a very common strategy that is used in a lot of cancers, but has been used in the past as one of the first strategies. Coming to the parts which are probably the most interesting now, because these are really used in the clinic and these are really rather recent, so it's really actively applied now, which are immune checkpoint inhibitors. And this is also where, again, these two won the Nobel Prize. So this was the strategy to what they developed or found basically that when you have a tumor cell and it communicates with a T cell that's actually coming up to kill them, basically the tumor cell has developed certain receptors on the surface that are binding to T cells and they're stopping them from killing the tumor cell. Basically, that's one of the findings they had. And if you use an antibody against this receptor, basically you block this connection. The T cell can recognize the tumor cell as a threat and can kill it. So basically, this receptor here is stopped. They block it, that's what they call an immune checkpoint. And they can block it, they can inhibit it. And for that one, the T cells are able to kill the cancer cell. PD-1 is probably the most prevalent or the most known checkpoint by now, because this was also connected to a lot of clinical trials and now also in the clinics. This is a therapy that is often given to cancer patients. CTLA-4 is a similar working mechanism. It works on a slightly different cell type, but basically here the interaction which is stopping the T cell from working is between antigen presenting cells, which are dendritic cells, and the T cells, which is sort of basically blocking this function of the T cell. It keeps it inactive, and if you block this connection, the CTLA-4, it gets active and it can kill the tumor cell. So this is the main idea behind these immune checkpoint inhibitors. You inhibit a certain crosstalk between cells, which enables the T cells to be active and then kill the tumor cells. What is done is often that this therapy is given in combination with a normal chemotherapy. So the T cells are active, they're already starting to kill the tumor. But when you give a chemotherapy, it's a more rapid process. So it's often a combination therapy of chemotherapy and an immunotherapy, or other different types of therapies that are given in the clinic. So now there are a lot of different checkpoints analyzed. I think there are now more than 20, 25, 30 checkpoints that are now actually investigated, which can all be blocked, and then they are all sort of promoting the tumor cell killing function. There are certain challenges connected to this, but I will come to this later, because this is a general challenge in immunotherapy. And also when you look at some clinical data or some market data, basically when you talk about immune checkpoint inhibitors and the market size, you can also see that this is really a therapy that is now coming up more and more, predicted to be huge market shares in the next 10 years. So basically working on this now, a lot of companies are now picking this up and working on this in the clinic. So it's a very promising strategy to fight cancer. Another promising strategy is adoptive cell transfer or adoptive cell therapy, which is actually very cool. So basically what is done here is that you use your body's own cells to genetically modify them and put them back to the patient. How does it look like? I think the most well-known adoptive cell transfer therapy is CAR T cell therapy. You might have heard of it. But basically, the cancer patient is donating blood. Out of this blood, you have the T cells are isolated by different mechanisms. Then in the lab environment, these T cells are genetically modified to express a certain receptor, which is in this case the chimeric antigen receptor, CAR. So these are CAR T cells. In the lab, you can easily grow these cells into the millions. And then these genetically modified T cells are given back to the patient. And then these CAR T cells bind to a receptor on the surface of the cancer cell, which in this case for CAR, it's CD19. They can recognize this, bind to it, and actively kill the cancer cells. So this whole strategy is amazing, that you technically take the patient's own material, you modify it in the lab, and you give it back to the same patient. And usually this process is done in about 2 weeks. So you can imagine this whole lab work is very fast. I think it takes roughly 1 week to grow them, to modify them, and then it's given back to the patient. And this is a very, very promising technique because it can also be used for very solid, like very stiff and large tumors. So it's a very promising technique for the future. It is now facing, of course, the challenges that you have patient material, you have to work in very, very controlled environments. It needs to be completely sterile, and you have to give it back to the patient. But the advantages, of course, when you use the patient own material, it reduces the side effects because it's the patient own cells. The body doesn't recognize them as a threat. So basically, this whole process is comparably safe if you use the same cells. Now, some companies are now continuing that research in a even more impressive way, I should say. One of these companies is Imetics. It's a German company, which I got in contact recently. And basically what they're doing is they have a cancer patient. They take a part of the tumor. They look into what are the antigens found in the tumor of this specific patient. So by now they have analyzed more than 200 of these markers for the patients. It's very impressive that they sort of have a library of different markers they can now use for their therapy. So they match basically this. They have a cancer patient. They match. Okay, the first two they're not matching, but the third one, the third antigen, is really specific for this cancer patient. Then based on this antigen, they genetically engineer the T cell, and then they again expand them and give it back to the patient. The advantage here is that these people are then really having a personalized medicine that is really specific for this cancer patient, and can be used also very, very fast. So they do this also in 1 to 2 weeks. And they have a very personalized medicine, which is now, I think, in a phase 2 clinical trial. So they're really bringing it to the clinic now. This is a bit how it works. You have the cancer patient, you have a tumor cell with a very specific protein on top, and this is the engineered cell that they genetically engineered, and then they bind to the tumor cell and they kill it. That's the whole strategy. A slight variation of this adoptive cell transfer, which is a bit more rapidly in the clinic, are sort of what they call bispecifics. It's a sort of linker protein, so you don't need to engineer the whole T cell, but it's also sort of links the T cells which are already there to the tumor cell by having a linker protein that they can still attack the tumor cell and kill the tumor cell. So there are now different strategies. You can either use this biological protein complex, which is then linking the T cells, or you can directly engineer the T cells to kill the tumor cells. So it's a very novel, cool approach to get rid of cancer, basically. And also here, if you look at the market shares, in the future, it's predicted to be much more coming. So really the checkpoint inhibitors and T-cell therapies are really growing and coming up in the next 10 years. Coming to the final step, and this is something I work with personally as well, are cancer vaccines. Basically, what it all started, and probably maybe that's the only good thing about COVID, is that these cancer vaccines are now pushed to the clinic because the companies producing the COVID vaccines, which are Moderna or Pfizer, BioNTech, now have the funding to push these cancer vaccines. It's the same strategy that they're using as in the COVID vaccine that I think we all got. And now they're really pushing these cancer vaccines to the clinics. How does it work? I will go briefly through it, but basically you have your cancer vaccine. This can be an mRNA, like in the COVID vaccines. This can be a protein. This can be a peptide. These are taken up by the immune cells. They sort of train your own immune system, the T cells and the B cells, against this vaccine that is often then considering one part of the tumor. These trained immune cells are infiltrating the tumor, killing the tumor cells, and also this killing of the tumor cells is releasing more and more antigens, which then again train the immune system to detect these antigens. The tumor is killed in a sort of circle. So the vaccine is actually given as a starting point to kill the first tumor cells, and then this whole circle is starting that actually these antigens released by the tumor cells are recognized by the immune system, and they start the whole process all over again. What is important to mention here is that these cancer vaccines are not given as a sort of vaccine that we know. It is hard to give it as a sort of preventive vaccine, so you cannot really give it to a healthy person that this person does not develop cancer, because for this, cancer is too individual. It's very different for each person. But it's already given when you have a tumor as one of the treatments, or you can imagine if you had a cancer, it got resected surgically, and then you give this cancer vaccine treatment, and it all sort of kills the remaining tumor cells which are still there. So that was a bit of the cancer vaccine strategy. We see the same a bit summarized here. Not all of it is interesting, but basically what we see, it can have different origins. mRNA is now the most well-known because this is also in the COVID vaccine. But you can also use DNA, you can use proteins, you can use peptides. Some approaches are also using parts of a tumor. So they actually take some tumor cells of the patient, they kill them, and then use these killed tumor cells as vaccine. So there are a lot of different strategies coming up in this region, in this strategy. So it's a very cool strategy. It's now not on the market yet, but I'm pretty sure it will be in the next 5 to 10 years. Yeah. So when we talk about cancer immunotherapies, so these were all the strategies that are available on the market. What you might have noticed is that all of these strategies, or most of these strategies, are focusing on this part of the spectrum. So currently, all of the immunotherapies that are available are actually working with the adaptive immunity. You train your T cells to recognize cancer, and they are killing it. Now, what is more and more coming up in the recent years is that also immunotherapies work with these part of the immune system, sort of the overlap between adaptive and innate immunity. So some of these cells, which belong to the innate immunity, are now also modified to kill cancer. How does it look like? Now, it's very crowded here. But basically, you have natural killer cells that are now modified to kill the cancer, also CAR natural killer cells. They work with the same receptor. You have macrophages that do the same. But basically, these cells are all also capable of killing the tumor very much. How does it look like in specific? Basically, these CAR natural killer cells, so natural killer cells, as they are called, are very good in killing, in this case, tumor cells or pathogens. And they are also modified with this chimeric antigen receptor to target the cancer cells and very specifically kill them. And you can see actually here a natural killer cell, which is green-blue. And here you can see a dying cancer cell, which is basically this blob structure, which is basically at the point of dying. A different strategy, which is now coming to a bit of our own work, is to not work with the natural killer cells, but to work with macrophages. So macrophages are part of the immune system of the innate immunity that we focus on. And basically, what we have here is that we have a couple of macrophages that we have different strategies for them. And basically, what we can do with our work is we can inhibit the function of these macrophages so that they cannot promote the tumor anymore. They cannot promote the tumor suppressive function anymore. They can directly kill these macrophages, which is a bit of a tricky strategy because killing cells can often be more worse than inhibiting or programming them. Or the interesting part is we can reprogram them, which is basically using the macrophage and then putting them into a state that is actively fighting the tumor. Now, all of this work has been published, so that was very nice. And how does it look like in vivo? So this was in mouse models. And basically, the strategy of killing the macrophages, we see that the tumor is nicely reduced here in this greenish line. But especially like this reprogramming, so making these macrophages kill the tumor by themselves, was rather effective. And now we are applying more strategies to look into that. And a follow-up of this, which is done by a different group from of Willem Mulder. It's a so-called trained immunity, that you inject something into the body that goes right away into the bone marrow, so the origin of your macrophages. And basically in the bone marrow, so really the site of where the macrophages are formed, you already train these macrophages to kill the cancer cells. So this is a very early approach that you sort of at the start of the whole process, you already train them to kill the tumor. So this is a very interesting approach, very promising. And yeah, I think they also started a company with this, which might be coming soon to clinical trials. All right, need to drink something. Yeah, so these are the strategies for the innate immunity which are now coming up. But as I said, most of the things which are in the clinic are more on the adaptive immunotherapy. So working mostly on T cells. Now coming to a different part, which are other diseases. There are not that many which are currently treated with immunotherapies. But I picked a few that work with the same ideas. One of them could be diabetes, which is now explored. So basically what happens in diabetes, what happens in the pancreas is that you have beta cells which are releasing insulin. One problem with diabetes is that your own immune system is sort of recognizing antigens found on these B cells, and they start an immune attack for some reason. So this is some issue in the body, that these T cells are attacking your own pancreatic cells. And as a result of the dying beta cells, there's a lack of insulin, which is the problem of diabetes. And some strategies for immunotherapy are now working on actually inhibiting this killing of the T cells. So the sort of, you can imagine if you have a blocking, like an immune checkpoint blocker here, that you might be able to stop this diabetes loop of the cytotoxic T cells killing your beta cells. And there are also other autoimmune diseases which are focusing on the same idea. Another disease which I think Professor Kuliah is happy about is that looking at liver fibrosis, there are also some strategies that are now working with immunotherapies, where basically you give in the earlier stages of liver fibrosis, before it reaches really the stage of no return, basically of cirrhosis, you give what they call vaccination. What is happening that you train your own immune system to fight fibroblasts, which are actually the main driving forces of fibrotic diseases, as the name implies. And basically at this stage in the liver, for example, you treat them with the vaccination, you target these fibroblasts, which are overproducing matrix, and you block this whole liver fibrotic cycle. This is a very new strategy. So I think actually immunotherapy to kill fibroblasts is hardly used so far, even in the academic part. So that's something I found which was very, very interesting to see, that also now in liver and lung fibrosis, people are using the strategy to target specific cell types. And then one other disease, which is also sort of autoimmune disease, it's a pemphigus. I'm not sure if it's pronounced right, but basically what your body is doing in this disease, you have certain proteins connecting your skin. And in this case, the body is producing antigens against these proteins. So you get sort of like opening in the skin. You get like lesions of your skin, which are very hurtful, which are bleeding. And now there are people working on T-cell therapies to kill these antigen producing cells. So basically these antibodies which are produced to open up your skin are not produced anymore, and they are sort of trying to kill them. Yeah, so it's very interesting that besides of cancer, there are certain strategies, but there are not that many. It's mainly focused on cancer. Coming to the last part, I think being good in time also. There are certain challenges coming to it. I will broadly mention them because of course, there are always challenges with all therapies. But one challenge that especially cancer holds, which is a huge problem in the field, is the underlying cancer biology. What happens within cancer is that all the different cell types that I also showed earlier in these graphs, they are all interconnected, they're all working together. And all of these cell types are technically inhibiting the immune system. It's not only the cancer cells, it's also the fibroblasts, it's also other cells, macrophages, macrophages, they're all in this functioning cancer inhibiting the immune system. So when you really want to get your immune system to work again, you need to target one. Yeah, you need to target not only one of these cell types, but technically all of them, because they can all have this immune suppressor function. So it's a very complex environment. It's a problem that I'm not sure if it can ever be solved, because it's such a complex environment. But basically, the immune suppression is not only one cell type, but it's the whole interconnective network that you need to target to suppress to get it done. One problem is also when we look at cancer cells directly. A cancer cell can mutate; they can have changes within the cell. So not every cancer cell might be expressing the antigen you're targeting. Not every cancer cell type might be, for example, expressing PD-1. So if you use a PD-1 inhibitor, it might not be effective. On the other hand, it can also be that some cancer cells have an increased expression of PD-1 that you cannot even add enough treatment for them to get rid of it. So basically, this mutation of cancer cells directly is a huge problem. And the same goes for the whole mutation of a tumor as a whole. Because in tumors, the problem is you have different cell types. Within this tumor, there are some tumor cells that you can kill, but there will be always some tumor cells that are resistant. And then what can happen is you can kill the tumor cells which you are able to kill, but then this resistant cell type has enough space to grow, and you actually have a completely new tumor where you need a completely new strategy to kill it. So this whole tumor evasion, this escape, is a huge problem, because it's very hard to kill the whole tumor as a whole cell, all the cell types. And usually what will happen is that with some of the resistant cells, they overgrow and take over, and then you need a completely new strategy. This is also the case why some of the tumors always come back. Even if you treat them, you get rid of it, and half a year later, it's back. So this is really a huge problem. And yeah, it's a very biological problem. So it will be very, very hard to solve. Let's see. The next problem, and that's really a problem with immunotherapy, is very specific for these ones, is basically what you do with an immunotherapy. It doesn't matter which one you use, but you change your immune system. In this case, it's good to fight cancer, but if you change your immune system, it can also have a lot of side effects. And something what usually happens if you have an immunotherapy, at some point, if you really overdo it, your immune system is sort of causing a cascade to be very, very active. And that can lead to a lot of inflammation. So basically what you can see here, myocarditis is the inflammation of the heart, inflammation of the liver hepatitis, of the skin, you have dermatitis. So once your immune system is overly active, the risk of getting inflammations is very high. And the problem here is inflammations can lead, for example, in the liver to liver fibrosis. So you get a very fibrotic liver. So this immunotherapy has always been very, very specific to each patient, and it cannot be overdone. So at some point you develop so much immune response that your body cannot handle it anymore. This also goes for any disease you had. So you can imagine if you have a cold and you get an immunotherapy, your immune system is already active, it gets overly active. That is a problem. So it needs to be very, very specific for each patient. One problem that we're also focusing on to reduce actually the side effects is the targeting of the immune cells. This is also one that we recently published. But basically, there are specific receptors. This is a scavenger receptor on immune cells, for example. And you can develop targets that actually really bind to this receptor. What we did in the past is that what actually Jay did in the past, he developed this PAPC receptor. It's a lipid, which can specifically bind to the scavenger receptor, and then in form of a liposome, which can carry a drug, it can be taken up. In this way, you can target specific immune cells and sort of reduce the side effects of it. Of course, one of the challenges here is also that a lot of targeting strategies that work for usually cancer nanomedicine are very tricky to target immune cells with, because usually when we target cancers directly, we want to evade the immune system, and now we want to target the immune system. So there are a bit of tricky mechanisms to target immune cells right away, but they're not more and more strategies to come up that actually really allow binding of any liposome or nanoparticle to the cell membrane so that they can specifically be taken up. Coming to already one of the last challenges that I want to talk about, it's basically that we work a lot in pharmacy and cancer research with preclinical models, which are often mice or rats. The thing here is, you can imagine, if you want to work with immune systems or immunotherapies, you need an animal model that has a functioning immune system. Oftentimes models in cancer research do not have that. They are immune suppressed. So we already need a very specific type of immune system mice. And one of the problems is that the immune system of a mouse is nearly completely different than of a human. So even if you test an immunotherapy in a mouse, it's hard to predict if it's working in a human. So there are very, very complex strategies to actually genetically engineer mice that they express human cells. There are strategies to get the immune system of a mouse, get rid of it, transplant bone marrow from a human patient to get a sort of humanized mouse hybrid. It's a very, very complex field. People are often going to monkeys very fast, there's also a bit of an ethical concern. But yeah, these preclinical models, especially in immunotherapies, are very, very tricky because the immune system for every species is very, very different. One alternative that I was also working on personally in the last years are these sort of alternative models for drug testing, which can be spheroids, which can be organoids, which can be organ on chip. Talking about them would be a completely new lecture, to be honest. But basically, these are 3D cell models that you can culture as a sort of an alternative for animal models. That's the idea. And with the FDA, so the US drug agency, recently allowing these models to be used as an alternative, even if you want to really develop something to the clinic, this might really be a chance to find ways of having a preclinical model that does not face the challenges of these animal models. And I think, by that I'm done actually. So it was a very broad overview of immunotherapies. These are all the people that I have been collaborating with in the past, from Twente, also from Harvard, and a lot of different universities. And yeah, thank you. Thank you for listening to me. And sure, if you have any questions, then I will try to answer them.
Okay, thank you, Marcel. Nice presentation. Yeah. Hello, Marcel?
Mm-hmm.
Are you still there? Yes.
Yeah, I'm here.
Yeah, good presentation. Yeah. In the chat, there are two questions. First, from Mrs. Henny. Yeah. Will you explain one more regarding the role of macrophages in immunotherapy?
Yeah. So technically, macrophages are they are considered part of the innate immunity, although they are sort of on the overlapping part of innate and adaptive immunity. Basically, the idea behind it is that macrophages are very, very good in eating up pathogens. They're the cells that are eating up tissues, dead cells. And in this case, they can be used to sort of eat up the cancer cells if they are trained or modified to do that. One strategy that we, in this case, use is that the scavenger receptor that I was talking about on the surface of cancer cells is also expressed on dying cells. So when cells die, they express this receptor, and this receptor is recognized by macrophages that this cell has to be eaten up. So in this case, macrophages can be used to eat up cancer cells. But it is not as easy as the current immunotherapies that are there, because it can be very, very tricky to train them. And it is these macrophages are usually not traveling a lot throughout your body. So T cells are constantly circulating throughout the body, but macrophages are rather local, and then you need to locally train them to eat the tumor. So the strategy of T cell is more used because it is sort of more moving throughout the body. Yeah. I hope that answers it.
Okay, any feedback? Mrs. Henny. Actually, Mrs. Henny is my colleague here, Marcel.
Sorry.
Mrs. Henny is my colleague here. Is to check about immunology, so is interested in this topic.
Yep.
Okay. Is any feedback, Mrs. Henny? Henny?
Okay, I think it's enough. Thank you for your answer.
Okay, sure.
Yeah. Okay, actually there are two more questions in the chat, but I will give speak first, Marcel. There are two students that raise hand. Maybe the first for Alfaikar, please, Alfaikar.
Thank you, Mr. Tentang. Good evening, Mr. Marcel from Indonesia here. It's now 8 p.m. here. I don't even know what time is there. What time is it there?
Now
Just
it's
remember
3.
that.
Yeah.
I just remember that I've heard so many news from that if someone discover cancer therapy, yeah, CIA will come to you. But that just means. May I ask you something? Nowadays, so many research about cancer treatment strategy. Yes, in the past, everybody think that cancer can be cured, but now it seems possible step by step. But from now, is there more evidence as far as you know, maybe like randomized control trial about the cancer immunotherapy that will be market someday, and perhaps that will be a good news for us. And that's the first question. The second is, just now I heard for cancer vaccine. I just want to know how potent is that. And the third question is, I'm so interested. Is it possible to discover immunotherapy for autoimmune disease? Yeah, that's my question.
Yeah.
Thank you, sir.
Yeah. So maybe to go backwards. So the last one, cancer immunotherapies for autoimmune disease. There are some. They are now developed. It depends a little bit on what cells are involved. So, for example, for diabetes, you can imagine there was this checkpoint of the T cells killing the beta cells. And if you then have a checkpoint inhibitor, you can stop this process. So there are some strategies that are now used to treat autoimmune diseases, but they are now coming up. I think it's also a bit of hype that a lot of cancer immunotherapies are now coming up. So there is money available to research others. Cancer vaccines are, they are called vaccines basically because of the mechanism they're using. They're not really vaccines that train your body if you're healthy to not develop cancer. Maybe in the future that would be perfect, but I doubt that this will be possible. But these cancer vaccines are mostly the idea if you had a tumor, it got resected, it got removed, and there are still some tumor cells remaining, that the immune system is trained to detect these remaining tumor cells and kill them. That any metastatic site, so any metastasis that was there is killed. That is, I think, now the main idea of cancer vaccines. And I have to ask for the first question again. It was a long one, sorry. So what was the first one?
Is there more evidence, maybe like randomized control trial about the cancer that we hope that will be marketed someday? Like that you tell is about the mechanism of immunotherapy that it's so amazing to it's very good thing, but is there like clinical trial, randomized control trial
for immunotherapies, clinical trials, or?
Yes, for human, not for animal like that.
Yeah. So especially these immune checkpoint inhibitors, they are in the clinics. So they're not even in trials, they are now used in patients. Often in combination with other therapies. So technically, when a cancer patient comes to the clinic, it's not only one treatment. They don't only get a chemotherapy, or they don't only get one treatment. Usually it's a combination of different treatments. And now these immune checkpoint inhibitors, they are combined with chemotherapy. And a lot of these adoptive cell therapies are now in clinical trials, so they're really far in late stage clinical trials to come to the market. Yeah, so they are really used in patients, and they see they are really well doing in patients. Yeah.
Hello? Enough? Okay.
One cell type, that's the thing. So oftentimes these treatments which are toxic to cancer cells cannot even reach them. And then you need to treat the whole. So when you're able to penetrate this solid tumor, these immune cells can kill. And oftentimes the drugs which we use in nanotherapies, they are not really able to go into it. They just cannot reach the cancer cells, but immune cells can. So that's why it works slightly better than what we are doing now. Yeah.
Thanks, sir, for your presentation, for your answer. I'm too amazed and appreciative. Thank you.
Okay, good. Thank you. Right up. Next question, I think is Maureen. Is
First of all, I would like to thank you for the presentation today and for giving me the opportunity to ask. So I'm Maureen and I have two questions based on your presentation today. The first question is, one of the steps for engineered therapeutics is having the 3D bioprinted model step first before going to the preclinical models. What exactly is the purpose of having the 3D bioprinted model? And for the second question is, has immunotherapy been widely used worldwide? Because personally, I haven't really encountered one and I'm not really familiar with it. Thank you.
Okay. I think we have to go to the second question first. I am not sure how worldwide it's used. So I know in Europe and in the US, it's very common in you're looking at clinical trials. So there are a lot of clinical trials running now, but there are also a lot of companies doing it here. So only in Germany, there are really a handful of companies really focusing now, all of them, on these T-cell therapies. BioNTech, Pfizer is now using the funding they got, but the money they made with the COVID vaccine to push their cancer vaccines. So I think especially in US and in Europe, it's now coming a lot. I'm not sure about the Asian market, I have to say. And about the first question, using these 3D models, so that's actually my PhD, to be honest. One of the problems that we faced when we were developing new drugs is that if you have a normal cell culture, which is in 2D, you have a well plate, you have usually one cell type that you treat. When we use that drug, which works in a well plate, oftentimes it doesn't work in an animal, because the whole system of an animal is way too complex to be mimicked by just a well plate. And by these bioprinted models or organochip models or organoids, these are all the same way. You create an environment which is more complex, which embodies a lot more cells. So not only tumor cells, but also immune cells, for example, to test your drug in. And what we now do in our group is that we see that the drug works in a well plate. We test it in a 3D model. And if it doesn't work, we go back to optimizing it. But if it works also in 3D, we go to animals. So in the end, it's an in-between step that allows us to reduce the animals we need and allows us to optimize our drug very rapidly, because animal studies take a lot of time. And such a model, you can do it way more rapidly before you go to animals. So that's the idea of these models.
All right, so thank you for the answer.
Yeah, you're welcome.
okay, thank you, Maureen. Next, Marcus, Ratu Akima, please.
Thank you for the opportunity. First, I appreciate about your presentation. I have question for this discussion. First, what do you think about cancer immunotherapy in the future, considering the current limitation of cancer immunotherapies as safety risk and resistance of many drugs? And second, among all challenges that you explained, what is the biggest challenge for cancer immunotherapy and why is it so? And third, that you explained about vaccine cancer. In your opinion, how much it is potential to be immunotherapy for cancer, and if it is it not so potential, how to optimize or technique to get better vaccine cancer? Thank you.
Yep. Good questions. I think talking about I think the cancer vaccines and immunotherapies in general are facing rather the same issues or the same challenges. When I look at the main challenge, which especially in cancer, is the cancer biology. It's something that we cannot change actually, but every tumor for each patient is unique basically. Every tumor develops a different. Every patient develops a different tumor, and having one strategy that fits all doesn't work. That is also why some of these approaches, which are very interesting, if you look at cancer vaccines or on cell therapies that are taking a part of the patient's tumor. So what happens is, for example, they take a biopsy of the tumor, they analyze it, and they match the markers with their therapy. So it's a very personalized therapy for each patient. I think this is a very, very nice way of working around this biology, because if you have this personalized way, it has a higher chance to work. This is also why strategies like chemotherapy or other therapies, which are very standard, for some they work, for others they don't. And with this personalized one, it might be better. I think this is probably the biggest challenge, that we need a treatment that works individually for each patient. And looking then at the risks behind it, or maybe one more thing, also the one challenge is these mutations within cancer. So usually you don't have one cancer cell type. You maybe if you're even able to kill 95% of all cancer cells, 5% might be resistant, and then these 5% have enough space to grow, and they overgrow, and then you need a new treatment. So I think this biological problem will be there, and I also doubt that we can ever solve it completely. The good thing about these immunotherapies and cancer treatment is that usually if you get a resection, like I always said, if you get the primary tumor removed and you have cancer cells left, if you train your immune system to detect them and kill them, you have a good chance of getting rid of the tumor, because then it's not that much cells. You usually have these metastasized cells are not that mutated yet. So if you have this really good time point of killing them, you are really, really good in getting rid of it completely. One problem that I also talked about are the side effects of altering your immune system. You're technically playing with something that is there to protect you, and you're sort of you can trigger an autoimmune response where the immune system is overactive and targeting your healthy tissues. And this is one of the major side effects that immunotherapies have at the moment, that at some point the immune system is overly active and it targets your healthy tissues, because some of the receptors which are used in immunotherapy are also there in healthy tissues, very, very low, but they are there. So, and then if you target, like, for example, your heart muscle, you get problems with myocarditis, like the inflammation of the heart or the liver. So this is something that, this is also why immunotherapies are used, but there is a limit to the times you can use it. So you need to treat them, and they need to see, or you need to have a break. So it's a very good question about this treatment strategy that you have. Yeah. Okay.
Any feedback for this?
For the answer.
Sure.
Okay, thank you for this. Unfortunately, I missed the question from Vela and Dava. Maybe someone can read in the chat.
Yeah, I can also read it. In Indonesia, immunotherapy develops on mostly take nature. Hey.
Hey, Marcel.
Hey. does the use of nature as objects of immunotherapy research also exist in new countries? If there's not, how is development of immunotherapy in there? That is a good question. So there are natural sources of treatments. So for example, if you have strategies that we use for macrophages, it's a sort of natural source, but we use a part of the membrane of bacteria. So it's technically natural, but not really nature, not really plant-based or anything. But I'm not aware of any clear natural products that are used. But there are strategies to use bacteria-based or really not only synthetic modifications. Of course, if you talk about genetic modifications of cells, then this is a genetic modification. And then regarding the current development of immunotherapy, based on your opinion, what is the most interesting thing to develop from the existing immunotherapy concept? I think the most interesting one that I see at the moment is these engineered immune cells. So if it's T cells or natural killer cells, which are engineered in a way that they're very personalized. So you take a biopsy of a tumor, you screen this tumor for the markers which are relevant, and then you engineer your T cells in a way that they are only targeting these cells. So companies like Immatics are doing that at the moment. They're now in clinical trials as well, so they're already treating patients. And then you have a very personalized immunotherapy. And in this way, you might be able to also evade the side effects by it, because it's very, very specific to the tumor cell. I think this is probably the best or most promising way of getting it to the clinics and to the patient. And then I think the other question, I have a question about the formulation and delivery of cancer vaccines. Are there specific techniques or technologies being utilized to optimize the formulation and delivery of cancer vaccines to enhance immune activation? Yes. Basically, all the strategies that were evaluated for Corona, the COVID vaccines, which were mRNA, which is now the best one or the most used one, are also the same for cancer vaccines. So people are using liposomes, people are using virus particles, people are using polymer nanoparticles to deliver mRNA, to deliver a protein, to deliver a part of the tumor, to enhance the immune system. So it's basically the same strategies that are there for flu vaccine, for the COVID vaccine, are now evaluated for the cancer vaccine. And what is pushed now are mostly liposomes with mRNA. The reason for that is that the companies which are now doing it are based on these techniques. It's also a bit a question where the money comes from. Yeah, but I think these liposomes and mRNA are most likely the ones which are reaching the clinical trials the soonest. Yeah. Okay.
Any feedback, Dava, Stella? Okay. Is there any question from students? Do you still have time, Marcel?
Yeah, I have some minutes.
Okay, some minutes. Marcel, might be in my mind, might be some of students here is still doubt about this macrophages reprogramming. Can you explain about macrophages reprogramming?
Yeah. So that's very interesting for you, of course, as well. So basically, what we have in cancer is that the macrophages, which are there, are they should kill the cancer cells, but they don't. They are sort of brainwashed into supporting the cancer cells. So local macrophages, or the macrophages coming from the bloodstream, they go there and they are sort of brainwashed into a very anti-inflammatory type of macrophages, which means they promote the growth of tumor cells. This is known as, technically, some years ago, M2 macrophages, which are in cancer. So they're very anti-inflammatory and promoting the tumor. And now more and more strategies come up to actually reprogram these M2 macrophages to M1. And this is also what we did in this nature study, which was basically using a pro-inflammatory drug to push these macrophages to the pro-inflammatory phenotype, that they get their phagocytotic function again, and then they kill the cancer cells. And by doing so, they also have other ways of degrading the matrix, opening up the tumor. So we're now exploring a lot more things about only killing the cancer cells. But basically, if you have this macrophage killing the tumor and combining that with the chemotherapy, it works very, very nicely. Yeah. And then, what is also often done is these macrophage reprogramming and the combination with immune checkpoint inhibitors. So you activate the macrophages and you activate the T cells, and combined they kill the cancer.
Okay. I think Marcel time is almost finished. Yes, Marcel?
Okay.
Okay. Thank you very much, Marcel. Yeah, I hope we can meet again offline. Yeah. And
yeah.
I always looking forward to coming here. Yeah. And we can play together with my little one.
Yeah, sounds good. I definitely have to come.
Okay. Thank you very much, Marcel. So we have to close the class today. Yeah. Again, thank you very much, Marcel. I think this is the students very enthusiastic. I'm surprised that they give you. I think it's
no, were really good questions. Yeah. Some of them I cannot even answer because it's so new. That's
yes.
Yeah.
Okay. Thank you very much, Marcel, Stephanie, and enjoy your time and enjoy your work. Greeting to Lena and Jay and everyone in the Twente. Thank you.
I'll do that. Okay, good. Thank you. Yeah.
Yeah.
All right.